The development process and future prospect of proteomics. Proteomics has become one of the most effective methods for molecular markers of disease and drug targets. In cancer, Alzheimer's disease and other major human diseases, clinical diagnosis and treatment, proteome technology also has a very attractive prospect. Recently, there are new breakthroughs in the development of proteomics, and we have prepared several materials for you. Professor John R. Yates, an international famous proteomic expert from the Scripps Institute in the United States of America, was invited to attend the 5th Asia-Pacific Mass Spectrometry Conference in Beijing to report. In the first half of his report, Professor Yates described the development process of proteomics and the direction in the future. The evolution of shotgun proteomics. The development of proteomics is together with the development of protein multiplex technology. Over the past 100 years, mass spectrometry has grown exponentially. This progress can be attributed in part to innovations in the mechanical, electronic, and computer industries. But there have been occasional disruptive breakthroughs. It is possible the fundamental cause for the development of mass spectrometry. It is these subversive breakthroughs that make large-scale protein analysis or proteomics possible. Another important task of proteomics is to identify the function of proteins encoded in a gene sequence. Shotgun proteome technology allows people to quickly acquire information through some new strategies, including methods based on concept of linked crime, enrichment and identification of protein based on activity, whole cell or organelle analysis, etc. Identification of protein function. Regulation of biological system. The molecular structure to modify proteins is very various. These modifications are of a significant regulatory function, and some are merely altering the chemical properties of the protein without significant regulatory functions. Modifications with regulatory functions are usually reversible except for proteolytic process. Protein quantification. Introducing stable isotope label into the human and animal body is for measuring the final metabolic product of the molecule. Metabolic analysis is achieved by tracing isotope-labeled amino acids and isotope ratio mass spectrometry techniques. Metabolic stability isotope labeling is a very powerful method for studying animal biology. The overall animal labeling enables research topics to involve more complex systems than cell lines and can better reflect the biological mechanism of organisms. Stable isotope labeling of animal bodies allows people to use tissues or organs for disease research. In addition, tissues and organs are, in fact, a collection of many different cell types, and ultimately, the purpose of the study is to understand how the confluence of these cells exerts their function. in vivo labeling of the whole animal. For the shotgun proteomics, quantitation and identification strategies will produce a paradox. In a full mode, the identification of proteins in a complex system requires rapid scanning instruments and efficient chromatography to maximize the capacity of MSMS. The instrument should be able to quickly collect a peptide data, and then move on to the next new peptide. In order to accurately measure the difference between the two forms, the peptide segment quantification needs to collect enough data points. The speed and lasting. These two paradoxes led to a compromise on the quality of quantitative data, because the detection limit for peptide identification is more than the limit of quantification. Another problem is measuring the presence or absence in the quantitative experiments. In order to further calculate peptide measurement results, most of the software tools require the presence of the peptide segments marked by heavy and light isotopes. When the ratio of different labeled peptides exceeds 10:1, the quantitative efficiency begins to decline, and some major changes may be missed. Some unlabeled methods, such as spectral counting, can better determine a number of large changes, but their accuracy is not as good as that of labeled method. Future prospect. Protein complexes represent a higher structure in the cell. Determining how protein isoforms or modified forms influence the protein complex function or activity will be the next step of work. At the same time, we also look forward to the progress of mass spectrometry by science and technology, as well as the intense commercial competition. In order to provide a better tool for proteomics, the scanning speed and sensitivity of the mass spectrometer will be further improved. Link: https://medium.com/@contact_28268/ The development process and future prospect of Proteomics 2254A0FD1CCB. Thank you for watching.